Good evening. Welcome to Short Talk Session 7. Sorry for the couple of minutes delay. We've just trying to connect one of our speakers who will hopefully join us for her slot at the end. Really excited about this group of talks. I'm going to hand more or less straight over to Laureline. Do you want to start sharing your screen?
So thank you very much. I'm going to share. And I'm going to discuss a theory for the population rate of adapting neurons that allow us to understand how single neuron properties shape neural population statistics. And really what I'm going to discuss first is that there is a little bit of a rift in how people model the neural response today, whether they think about isolated neurons in slice, which are spiking, reliable, and modeled with more detail, or whether they're trying to capture the response of networks in vivo, where they often rely on rate equations with tuned connectivity. So the reason why it's possible to go with more details in slice is that it's possible to patch single neurons and to really control the current that is injected in the neuron and to record the voltage. And what you observe is that the single neurons are going to respond with these large spikes that occur at relatively reliable times with millisecond precision when you inject the same current. And it's possible to model this really well using models that decide spike times as a function of the voltage and the time of previous spikes. But when thinking about neural networks in vivo, in contrast, when using these rate equations, what people will often do is to develop, for instance, functional models where you have a network, a rate network, and you train it to return a particular output, for instance, an electromyogram if you're modeling, say, motor cortex. And you can compare trial average firing data with the model. Another way that people use rate network is for empirical descripting models. You want a network that is going to feed the data, and this is a pipeline that will produce firing rates from which spikes that are, as you can see here, very asynchronous, very noisy, are drawn. And the question really is, those red squiggles, those firing rates in those rate networks, how do they mechanistically relate to single neurons dynamics? And it's a question that has been studied in the past using theory, but there are some limitations, notably to take into account adaptation, and that's what I'm going to talk about today. And the reason why I'm going to talk about adaptation is that if you really want to have a neuron model that is going to capture the response of a patched single neuron, the way you model it is you're going to have, at least in some models, a probability of spike in a very small time being that is going to be proportional to a very small intrinsic noise, which is a very small probability that the neuron would spike in the absence of input, and a nonlinear function, here an exponential, that includes two different terms, including a filtering of the input current. And this filtering is a filtering through the membrane filter K. And this gives us a drive to this neuron such that after passing through the exponential nonlinearity, some spikes are being drawn. And what the spikes are doing also is that they are filtered through this adaptation kernel here, an adaptation filter. And these spikes, S here, what they do is at each spike time, when they get convolved by this adaptation filter, they decrease the probability of spiking of the neuron. So what this gives you is this very reliable spiking response. And if you knew what the current looks like in vivo, in principle, you should be able to understand how this single neuron then responds in vivo and how that could relate to rates. So the question really is, what is the input current in vivo, where we don't inject it, but it comes from the network? And a clue, because it's hard to measure those input currents directly, comes from already looking at the firing from simultaneously recorded neurons. And as I'm showing here, this firing in vivo, it tends to be very asynchronous. And on top of that, even though there seems to be some correlations at the level of the rate of these different neurons, once you condition the response on the population rate, there is very little correlations in between the spiking of two different simultaneously recorded neurons. It's very close to a random shuffle that would have no correlation at all. So this is the asynchronous state. You have this uncorrelated spiking across neurons, often in vivo, and that also happens in recurrent circuit. Modeling suggests that this is due to excitation inhibition balance, and that also implies that the total excitatory inhibitory current response when a population of neuron is stimulated externally is approximately uncorrelated across neurons. So uncorrelatedness in vivo is really what should be taken out from this slide. So then the question is, can we think of the rate as an average activity of a population of neurons? And what are the statistics of the input in this population in vivo? And what that means is that we have this population of neurons with different pyramidal neurons, I and I+1. There is also fast inhibitory interneurons in this population. Here we're going to take them so fast that the response is going to be assumed to be almost instantaneous. And we have different populations that interact with one another. And we want to try to understand the population rate in the black population here. And as I mentioned, really what that means is that we also have to try to understand what the input looks like in vivo. So we have these different groups of neurons, G, that project to each neuron of this population. And so the input is going to be the sum over the different groups of the synaptic currents that are being driven by spike trains from neurons in the presynaptic groups. And these synaptic currents, you can get them by convolution by the membrane filters that we talked about before, and this effective synaptic filter, which in our case is really this already balanced excitatory inhibitory response. And the sum of all of these currents invoked by the presynaptic spikes are the drive. And the question is, what is this drive looking like in the asynchronous state across different neurons? And I told you that there is some reason to believe that this drive is relatively uncorrelated between neurons. Mathematically, what that means is that if you have different presynaptic groups, K and H, with population rates RK and RH, which projects through these different neurons I and I+1, the expectation of the product of the currents that are evoked by these presynaptic spikes is more or less equals to the product of the expectations, which is themselves just depend on the rate. The consequence of that is that the drive D is this sum of many, many weakly correlated and identically distributed variables for each group. So it's approximately Gaussian across the population, and it has no time-dependent mean and variance. And what we want to try to know is what is the population rate that averages over the single neuron statistics over this distribution of drive in different neurons of this black group. And so mathematically what that means is we want to compute the expectation of this firing probability lambda. And in our models, there are two types of stochasticity for this firing probability. There is this intrinsic noise, which is very small, but which gives a little bit of variability in the response for a fixed drive. And then there is expectation over the drive itself, which changes across neurons. And for the first expectation, previous work has shown that you can have a very good approximation on timescales that at least are longer than the very short refractory period by having a slightly different adaptation kernel eta tilde that now convolves not the spike train, but the private spiking probability of the neuron. And all we need to do now is to compute the expectation over the different drives, which may not be as simple as one might hope. One can do a first naive attempt to compute this expectation by neglecting the variance of the drive across the population. This would be a naive rate type of model where you're just replacing the drive and the adaptation by their averages. And then you can compare what happens in simulations and in the model for different means of the drive and different variants of the drive that are different shades of gray and shades of cyan in the theory. And what happens is that the theory not only it strongly underestimates the rates because of this, it neglects this exponential nonlinearity, but also it's qualitatively wrong in the sense that different variances of the drive now give you the same response in the theory, whereas in the data, the firing rate increases when you increase the variance. A second attempt that has been suggested in the past is to just neglect the variance of the adaptation across the population. So the adaptation is the same as we had before, and now we account for the exponential nonlinearity, and we have a contribution of both the mean, the expected drive, and the variance of this drive. This assumes that the adaptation is much slower than all the other timescales, but this is not accurate given realistic adaptation timescales. Really what this approximation is saying is that you have these different neurons, I and I plus one, they have these different drives, they have these different adaptation shown in brown that correlate with their private drive, and you're trying to replace these specific brown drives with this average purple curve that's the same across neurons. And this is not going to work very well, as you can show here. Can see here, the firing rate is overestimated. And here you really have to compare the black curve with the dark gray curve, the gray curve with the medium green, and you can see that they don't match. So the third attempt, which is our contribution here, is to consider adaptation as a linear filter of the drive. What that means is that we can replace this neuron-specific firing probability here approximately by a linear filter, that gamma, that we can compute analytically of the drive D. And so what that means is that now the rate is the exponential of a convolution of this drive D, which is itself Gaussian. So it's an exponential of a Gaussian variable. The prediction of this already would be that the instantaneous rate distributions should be log normal. And this is indeed what we often see in vivo. And we can now look at what happens in simulations and in this equation here. And now we're getting a very good match across a larger range of mean and variance of the drive. The other thing that we can do is to look at the response as a function of time in a non-stationary regime. And you can see that this also works well. The red and the black are pretty close to one another, and a naive rate equation does pretty bad. Finally, the last thing that we can do is to actually express and work out this expectation here. And what we are going to find is that this gives us a response that has a mean-driven regime that's the same as the naive mean-driven rate model in attempt 1. And the time scale of this mean-driven response is relatively slow. But it also has a fast variant-driven response that accounts for both the drive and the adaptation. And these relative timescales, they don't depend on the specific parameters. And so they make prediction for the timescale of response that you can actually verify in the data. You can patch neurons or a single neuron and record its average response when you change the mean at a particular time of the input versus when you change the variance of the input at a particular time. And you can see that when you change the mean, you have both a slower increase of your response and also a slower adaptation. So this works relatively well. This is consistent with what we see in slices and in vivo. And what this means is that we were able to replace this big population of neuron with a rate equation here. And something that you can also work out is that this rate equation here is really equivalent to a system of coupled nonlinear differential equations, which looks like your typical rate models, except that now you have several variables and several different timescales for each population. So it's a higher dimensional rate model, a little bit like an LSTM in machine learning. And so what we have seen is that really, with this approach, we can understand how rates emerge when you're considering a network of population of neurons, and each of these population of neurons has a higher dimensional rate equations. And this also paves the way for asking whether these biologically derived higher dimensional rate equations may be beneficial when trying to train networks for specific tasks. And with this, I would like to thank you for your attention.
Thank you for that. It was really interesting and really clear. I'm afraid for timing reasons, we should probably move on, but people should feel free to either ask you a question with the tab or on Discord. You can go there and kind of continue conversation.